Hello, I'm Dr. Saferin presenting the poster Folate Prevents Autism-Relevant Phenotype Caused by Developmental Pyrethroid Exposure in Prairie Voles. Neurodevelopmental disorders such as autism and ADHD now affects 1 in every 6 children in the United States. We don't know what causes it or how to treat it. Pyrethroids are one of the most commonly used pesticide, and studies have shown that developmental pyrethroid exposure or DPE is a potential risk factor. Metabolomic studies in mice have shown that DPE alter folate metabolism in the brain of adult offspring, thereby suggesting a potential pathway for these effects. In this study, we hypothesized that DPE directly affects folate metabolism and that maternal folate supplementation can prevent these behavioral and molecular changes. In our study, we used prairie voles, which is a socially monogamous outbred species, therefore highly useful for studying autism-relevant social behavior in animals. We divided pregnant voles into 3 groups of control pesticide exposed, which is DPE, and a DPE group supplemented with methylfolate, which is also the bioactive form of folic acid. They were exposed to a low dose of deltamethrin and a high dose of methylfolate every 3 days throughout preconception to lactation. All our behavior tests were performed on the offspring throughout their lifetime for autism-relevant behaviors and organized into 5 major domains, including locomotor activity, cognition, social interaction, repetitive behavior, and communication, followed by molecular tests. In the locomotor domain, we see that DPE causes an increase in hyperactivity and overall movement over 24 hours, and maternal folate supplementation helps to prevent it. In the cognition test, animals learn over a period of 10 days how to get food when they are hungry. As you can see from the flat curve, DPE animals did not learn how to get food even when they were hungry compared to control and supplemented animals, and only 22% of DPE animals passed the acquisition test at the end of 10 days. This suggests that DPE causes deficits in the cognitive domain, which is prevented by folate supplementation. Social behavior is measured using partner preference and consolation test. All 3 groups showed natural preference towards their partner. In the consolation test, DPE voles had a reduced consoling response to their partner's distress compared to controls and supplemented animals, suggesting the preventative effect of folate supplementation. In the communication domain, we saw exposed animals had fewer and shorter calls. That was again prevented by maternal folate supplementation. For repetitive behaviors, we used marble bearing and observation test. DPE animals showed an increase in repetitive digging, rearing, and self-grooming, which was not prevented by folate supplementation. Finally, to assess the combined behavioral phenotype across all domains, we calculated individual z-scores representing their difference from the control group. We saw that DPE induced a consistent and strong phenotype across all measures as compared to controls, and maternal folate supplementation reduced this DPE phenotype to an intermediate value above controls. Folate metabolism is a part of the 1-carbon cycle. For molecular experiments, we measured total plasma and cerebral folate level and the expression of 6 metabolic proteins from the folate metabolism pathway. First, we saw that DPE animals had higher plasma folate level that was normalized by supplementation, and a similar trend was seen in cerebral folate level. This could potentially be accumulation of unmetabolized folic acid, which is not harmful by itself, but could be an indicator of poor folate metabolism. In our protein expression data, we saw that DPE animals had higher SHMT1 expression that was not normalized by supplementation and increased MTHFR and FOLR1 expression in the supplemented group. We performed a correlation between molecular and behavior results. Animals with the highest SHMT1 had the most repetitive behavior. Animals with the highest MTHFR and the lowest plasma folate level had the best cognition. Here we propose a possible mechanism that might be causing autism-relevant phenotypic changes in the animals. SHMT1 was the only target that was affected by DPE and not corrected by folate supplementation, similar to repetitive behavior. SHMT1 sits in a critical spot acting as a molecular gate for the flow of 1-carbon units to either DNA synthesis or DNA methylation. Therefore, increase in SHMT1 could be shunting away 1-carbon units from methylation towards DNA synthesis. MTHFR is critical in converting folic acid into its bioactive methylfolate form, and FOLR1 is critical in transporting folic acid inside cells for metabolism. Therefore, increased MTHFR due to supplementation could be helping to produce more bioactive 1-carbon units to be utilized in DNA methylation, and increase in FOLR1 due to supplementation could be helping to feed more 1-carbon units into the cycle, thereby balancing it between both pathways. In conclusion, our data shows that the most common pesticide causes autism in children. Methylfolate could potentially be a better alternative because it is the bioactive form of folic acid, hence would assist in proper neurodevelopment even in cases of poor folate metabolism. Thank you.